and found that there were 99 differentially methylated genes, or transcripts. And what I mean by that is that they were either all methylated in the dwarf and not methylated in the wild type, or opposite, they were all methylated in the wild type, and none were methylated in the dwarf. And what we found was 81% of those 99 were methylated in the dwarf. So that GNMT, that elevation in the DNA methyltransferase, and that suppression in S-adenosylmethionine was actually working in the way that we thought it would, excuse me, with regard to methylation. So the dwarf is more methylated on these genes in comparison to the wild type. So again, this is fairly new data, and we're just beginning to take a peek at these. And of those 99, one of those genes was GSTP. And so, as I showed you before, the expression of GSTP, the message expression, is significantly suppressed in the Ames in compared to the wild type. And that makes sense when you think about methylation. Primarily, methylation suppresses gene expression. And so, indeed, that's what we were seeing in these animals, and which was very exciting, except that we were hoping that all the GSTPs would be upregulated. And again, when you run to the literature, it tells you that GSTP is the only GST that's found to be methylated in mammals and in human, in addition to humans, and that it's actually methylated in cancer. So in prostate cancer primarily, and in some breast cancer cases, GSTP is methylated. So we, without looking at methylation in particular, we looked at another animal model because we still believe that growth hormone is involved in coordinating or regulating methionine and glutathione metabolism, which results in these differences in stress resistance and ultimately longevity. So we looked at an animal model called a growth hormone transgenic. So these animals have their overexpressed growth hormone. They have tons of growth hormone. They're huge. They look like small rats. And they live only half as long, so they live to 12 months of age. So normal animals live to 2 years. So these animals are extremely interesting at the other end of the spectrum. And we found that indeed GSTP expression was extremely high in these animals in comparison. So we're guessing, which we haven't run the assay yet, we're preparing that, to look at methylation in the growth hormone transgenic, and we should see the opposite if our hypothesis is correct. So I have it in quotes that the presence of high growth hormone prevents methylation. We have to determine that yet, but that's what it appears. These animals have much higher expression of this particular gene. So again, we're just at the very beginning of looking at this, beginning to look at what's going on with regard to the GSTs and stress resistance. And we know that the GSTs actually inhibit stress-activated MAP kinase signaling. There's only 2 known MAP kinases, ASK1 and MEK1, that are affected by this inhibitory action of GSTs. The GST mu classes block this pathway right at the very beginning here. We know that GSTM is really high in the dwarf, so they may have a greater break on stress-activated signaling at this point. But again, this is we're just hypothesizing right now. As you move along downstream in this pathway, you activate P38, or the junk pathway. And we know that GSTP from the literature blocks junk activation. And this particular gene is suppressed in the Ames dwarf. So what we figured out thus far, which is not very much, is that the relationships between thiol-activated proteins and these GSTs and the stress-activated MAP kinase pathway is extremely complex because there's lots of other signaling pathways that impinge upon these pathways, which result in either apoptosis or cellular proliferation, depending on what's going on. We know that junk functions within the overall context of activation status of all those other pathways. So you may have chronic junk activation, but it may lead you down a apoptosis pathway or cellular proliferation, depending on what's going on with all the other pathways involved. It's very and it's also very cell type specific. So we believe that the lower GSTP expression in the dwarf may result from chronic junk activation because they don't have this break on activation of this particular kinase. And we do have a little bit of evidence downstream of that. And if you hit up hepatocytes from dwarf mice with hydrogen peroxide, they undergo apoptosis much more readily than wild-type animals. So it suggests that potentially that this may be the mechanism here, that they don't have GSTP, putting kind of the brake on junk activation. But that's the only data we have evidence for at this point. So further work is warranted, obviously, in this area. So, kind of in summary, what I've told you that is in the absence of growth hormone, such as the Ames dwarf mouse, you have an increase in methyltransferase activity, which leads to a differentially methylated DNA between Ames dwarf and wild-type animals. In addition, you have an increase in cysteine availability, which is used in making up a variety of different thiol compounds, including GSH metallothionein. And in the presence of growth hormone, methionine metabolism is lower or suppressed, so you have less methyltransferase activity, which obviously affects DNA methylation. I hope to tell you more on that story later. We also know that cysteine availability is decreased because GSH levels are lower, and this has an overall effect of decreasing your detoxification abilities and decreasing your ability to fight oxidative stress. So we think that growth hormone causes a shift in resources. So when growth hormone is present, there's an increased demand for those amino acids for growth. So the big picture leading back to the first slide or two that I showed you with that whole table of growth hormone mutants, if you think about stress-resistant mutant or stress-resistant phenotype in general, it's common among long-living animals ranging from yeast to worms to flies to mice. These animals, long-living animals, exhibit resistance to a variety of different types of stressors, including oxidants primarily, because that's what most people have looked at, but also heat, heavy metals, carcinogens, that type of thing. And we think that the growth hormone pathway itself, when it's activated, negatively affects both methionine and GSH metabolic pathways. So, again, we believe that growth hormone and maybe IGF-1 and insulin signaling are mediators of these stress-resistant pathways and the resultant longevity. The degree of growth hormone signaling, which I didn't show you, when there's different amounts of signaling going on, it's reflected both in changes in growth in these animals and in their metabolic, downstream metabolic effects and in defense mechanisms. And we think that growth hormone is involved in defense mechanisms not only through the expression of GST and methionine with regards to detoxification and antioxidant defense, but also through epigenetic effects because of the methylation differences that we see between these animals. So reduced somatotropic signaling or reduced signaling through the growth hormone pathway may allow for some metabolic reprogramming to go on, and it's shifting resources away from growth because there's no demand for protein synthesis, and shifting those resources towards stress resistance and cytoprotection. And that's it.
Thank you, Holly.